In the intricate symphony of life, lipids take center stage as the silent choreographers of energy, structure, and function. These remarkable molecules, including fatty acid, glyceride, and cholesterol, are the unseen architects of our vitality. Lipid metabolism, the intricate dance that powers our cells, holds the key to understanding how our body transforms food into fuel, builds cellular fortresses, and orchestrates the harmonious rhythm of biological processes. In this report, we embark on a journey into the captivating realm of lipid metabolism, where we decode the mysteries of how lipids are digested, transformed, and utilized in the body. From the vital roles they play in the energy production to their influence on our cardiovascular health. The world of lipids is a complex and fascinating tale that impacts our well-being every day. Join us as we unveil the secret of lipid metabolism and explore their profound importance in the magnificent machinery of life. First and foremost, before we proceed to the lesson proper, let me first give you the purpose and scope of this report. By the end of this video, we are expected to discuss the following. Number 1, overview of lipids, definition and classification. Number 2, lipid metabolism overview. Number 3, lipid digestion and absorption. Number 4, the lipid transport in the bloodstream. Number 5, lipid storage and utilization. Number 6, regulation of lipid metabolism. And last, the clinical implications. Now let us proceed. Lipids are a diverse class of organic compounds that plays essential role in the human body and in biological world in general. They are hydrophobic molecules, meaning they do not dissolve in water but are soluble in nonpolar solvents like chloroform, benzene, and organic solvents. Lipids are crucial for various biological functions, including energy storage, structural components of cell membranes, insulation, and signaling molecules. Moreover, lipids can be classified into several main categories based on their chemical structure and functions. First, fatty acids. Fatty acids are the basic building blocks of lipids with differences in saturation. This saturation impacts their health effects. Next one is the glycerides, which store energy in our bodies and are found in fats and oils in the diet. Next is what we call the phospholipids that forms the walls of our cell membranes with polar head that loves water and two nonpolar tails that repels it. Next is cholesterol, which is a vital lipid that serves as both structural component of cell membranes and is precursor for important hormones like estrogen and testosterone. Similarly, steroid with the same function as cholesterol that plays a huge role precursor for important hormones like estrogen and testosterone. Lipoproteins that helps transport lipids into the bloodstream, including HDL and LDL. Next is what we call the glycolipids that contribute to cell recognition and stability by combining lipids with carbohydrate groups. Sphingolipids are essential for cell signaling and the structure of cell membranes, encompassing various types like ceramides and glycophingolipids. Understanding the classification of lipids is essential for appreciating their diverse roles in living organisms, including energy storage, cell structure, signaling, and many other crucial functions.
And now, I will be discussing an overview about lipid metabolism, and I divided it into 4 parts to make it easier for you to understand. The first one is lipid digestion and absorption. Triglycerides, being substantial molecules, do not dissolve in water like carbohydrates and proteins. They tend to aggregate into large droplets when exposed to a watery environment, such as the digestive tract. To facilitate their digestion, the digestive process must first disintegrate these fat droplets into smaller ones and then enzymatically break down lipid molecules through the action of lipase enzymes, while the mouth and stomach contribute to the process to some extent. The majority of enzymatic lipid digestion occurs in the small intestine. The end products of lipid digestions are absorbed into the bloodstream and distributed throughout the body. This, too, involves special mechanisms because lipids are not soluble in water and cannot mix with the aqueous blood. The second one is the lipid transport in the bloodstream. Blood lipids consist of chylomicrons formed within the intestinal mucosal cells during absorption, as well as lipids derived from storage depots, such as livers and adipose tissue. Lipids are transported as lipoproteins in the blood, and they are also transported as lipoproteins due to their hydrophobic nature. Next one is lipid storage and utilization in cells. Triacylglycerols are the primary storage form of long-chain fatty acids, which are broken down for energy and used in the structural formation of cells. The job of chylomicrons is to deliver triglycerides originating from the digested food to the cells of the body where they can be used as an energy source or stored in an adipose tissue for future use. And how do the triglycerides get from the chylomicrons into cells? An enzyme called lipoprotein lipase sits on the surface of cells that line the blood vessels. It breaks down triglycerides into fatty acids and glycerol, which can then enter nearby cells. If those cells need energy right away, they'll oxidize the fatty acids to generate ATP. If they don't need energy right away, they'll reassemble the fatty acids and glycerol into triglycerides and store them for later use. And the last one is regulation of lipid metabolism. The process of lipid metabolism involves the bile that dissolves the majority of the fat that the body consumes into tiny fragments. Then the pancreas and the small intestine release lipase, which hydrolyzes the fatty acids into free fatty acids and monoglycerides. Only a tiny portion of fatty acids fully hydrolyzes to form glycerol and fatty acids. Dynamic equilibrium governs lipid metabolism at all times in order to meet the body's metabolic demands. Some lipids are oxidized continuously while others are produced and preserved as a result of metabolic pathways that the body employs to produce and break down the different types of lipids are the main topic of this chapter. And this will be further discussed with the next reporters.
The digestion and absorption of lipids present unique challenges. Lipids being sizable molecules typically lack water solubility. Similar to carbohydrates and proteins, lipids undergo fragmentation into smaller constituents for absorption. Considering that the majority of our digestive enzymes operate in water-based environments, the question arises, how does the body effectively break down fat to make it accessible for the diverse functions it needs to fulfill within the human body? Let's start over with the mouth. The act of chewing mechanically reduces food into smaller particles and combines them with saliva. In addition, cells located on the tongue produce an enzyme called lingual lipase, which initiates enzymatic digestion of triglyceride by separating individual fatty acid from the glycerol backbone. In the stomach, the blending and agitation process aids in the dispersion of food particles and fat molecules. Special stomach cells produce an additional lipase enzyme known as the gastric lipase, which further assists in breaking down triglyceride. The lingual lipase, which ingested along with food and saliva, remains active in the stomach. However, it is worth noting with these two lipase enzymes have relatively minor role in fat digestion, except in the case of an infant. The majority of enzymatic digestion of fats takes place into small intestine. As the stomach contents move into the small intestine, the majority of dietary fats remain undigested and form larger droplets. The liver produces and the gallbladder stored bile is released into the duodenum, initial part of small intestine. Bile salts have both hydrophobic and hydrophilic properties, enabling them to be attracted both fats and water. This property makes them effective emulsifier, breaking down large fats gullible into small droplets. Emulsification enhances the accessibility of lipids to digestive enzymes by increasing the available surface area for their action. The pancreas releases pancreatic lipase into the small intestine for the enzymatic digestion of triglyceride. This process breaks down triglyceride into fatty acids, monoglyceride, which consists of glycerol backbone with fatty acids still attached, and some free glycerol, cholesterol, and fat-soluble vitamins, on the other hand, do not require enzymatic digestion. The absorption of lipids in the small intestine. Following the breakdown of fats, including the production of fatty acid, monoglycerol, glycerols, cholesterol, and fat-soluble vitamins, this substance must enter the bloodstream for utilization by various cells throughout the body. Bile remains a pivotal component in this process. as bile salts cluster around the byproducts of fat digestion, creating micelles. These micelles aid in bringing fats close to the microvilli of intestinal cells, facilitating absorption. Subsequently, the byproducts of fat digestion transverse the membrane of intestinal cells, while bile salts are refused to continue their function of emulsifying fats, forming micelles. Upon entering the intestinal cell, short and medium chain fatty acids along with the glycerol can be directly absorbed into the bloodstream. However, larger lipids such as long chain fatty acid, monoglycerol, fat-soluble vitamins, and cholesterol requires assistance for absorption and transport to the bloodstream. Within the intestinal cell, long chain fatty acids and glycerides undergo assemble to create triglycerides. These, along with the cholesterol and fat-soluble vitamins, are then incorporated into the carriers called chylomicrons. Chylomicrons are substantial structures consisting of a core and made triglycerides and cholesterol, surrounded by an outer membrane composed of phospholipid and infested with proteins like lipoproteins and cholesterol. This outer membrane imparts water solubility, enabling it to navigate the aqueous environment of the body. Chylomicrons originating from the small intestine initially enters the lymph vesicle, which subsequently transport them into the bloodstream. Role of bile acids and lipoproteins in lipid digestion and absorption. Bile acids produced in the liver and stored in the gallbladder are crucial for digesting fats. Released into the small intestine during the fat digestion, they emulsify fat into smaller droplets, facilitating the action of lipase. Lipids digestion yields fatty acid, monoglycerol, and glycerol, which are absorbed in small intestinal cell and transport to the liver via portal vein. Lipoproteins, particularly chylomicrons formed in the intestinal cells, play a key role in transporting lipids, including cholesterol in the bloodstream. The process is vital as lipid being water insoluble require lipoproteins as a carrier proteins for effective blood transport.
Lipid transport. Blood lipids consist of chylomicrons formed within the intestinal mucosal cells during absorption, as well as lipids derived from storage depots such as liver and adipose tissue. Blood lipids are transported as lipoproteins due to their hydrophobic nature. Now let's talk about how lipids are transported as lipoproteins in the blood. Lipoproteins are lipid plus a protein. Because lipids are less dense than water, the density of lipoproteins decreases as the proportion of lipid to protein increases. Lipoproteins have a major role in lipid and cholesterol transport and metabolism. Lipoproteins are classified based on their density and composition. The main lipoproteins in the blood are called chylomicrons, very low density lipoproteins VLDL, low density lipoproteins LDL, and high density lipoproteins HDL. Now, let's learn about the main lipoproteins. They are called chylomicron, VLDL, LDL, and HDL. Chylomicrons are synthesized in the small intestine from dietary fat, and VLDL, LDL, and HDL are synthesized in the liver and small intestine. Chylomicrons enter the liver and are packaged into VLDL. VLDL is involved in the transport of triglycerol from the liver to extrahepatic tissues. LDL transports cholesterol, so-called bad cholesterol, to tissues, and HDL is responsible for reverse transport or removal of cholesterol, so-called good cholesterol, from tissues. The terms bad and good refer to the nature of transport. For example, the good cholesterol refers cholesterol being carried by HDL, which is meant to be routed to the liver for bile formation or excretion, thus leaving the body and that deposited like LDL into blood vessels. Next, let me explain atherosclerosis and the role of lipoproteins in cardiovascular health. Atherosclerosis is a complex, chronic progressive disease that affects large and medium-sized arteries. Atherosclerotic lesions are promoted by low-density lipoproteins and form from accumulation of fatty substances, cholesterol, cellular waste products, calcium, and fibrin in the inner lining of the arterial wall.
Lipid storage and utilization. Lipid storage and utilization in the body are critical processes that contribute to energy homeostasis, cellular function, and overall health of the body. Lipids, which include fats and oils, provide energy, insulation, and are essential components of cell membranes. Understanding how the body stores and uses lipids is critical for metabolic balance and overall health. Let's first tackle the lipid storage in adipose tissue. Adipose tissue, also known as body fat, plays an essential role in lipid storage. It is the primary site for lipid accumulation and long-term storage in the form of triglycerides. These triglycerides are made of three fatty acid molecules linked together by a glycerol backbone. Adipose tissue is found all over the body, including subcutaneous fat and visceral fat. A variety of factors influence lipid storage in adipose tissue, including dietary intake, genetics, and hormonal regulation. Excess dietary calories are converted into triglycerides and stored in adipocytes, which are specialized cells within adipose tissue. Insulin and other hormones promote the uptake of circulating fatty acids into adipocytes, allowing them to be converted into triglycerides. On the other hand, hormones such as glucagon and epinephrine stimulate the breakdown of stored triglycerides, releasing fatty acids and glycerol back into the bloodstream for energy production. Adipose tissue has both white adipose tissue, which is primarily responsible for long-term energy storage, and brown adipose tissue, which is involved in thermogenesis, particularly in infants and hibernating animals. Next is the lipid utilization in energy production. Lipids are an abundant source of energy. When the body requires energy, it can access and use stored lipids. Glycolysis, the breakdown of triglycerides into fatty acids and glycerol, is the process by which lipids are utilized. Fatty acids are then oxidized in various cells to generate energy, primarily adenosine triphosphate. Skeletal muscle cells and hepatocytes in the liver are the primary sites of lipid utilization for energy production. Fatty acids are oxidized in skeletal muscle during physical activity and contribute to sustained energy production, making them a significant source of energy during endurance exercises. During periods of fasting or low carbohydrate intake, lipids can be converted in the liver into ketone bodies, which can serve as an alternative fuel source for other tissues, particularly the brain. The liver is also involved in lipid metabolism, including triglyceride synthesis for storage, and the production of very low density lipoproteins or VLDL for lipid transport to other tissues.
Let us now discuss the lipid metabolism in the liver and muscle cells. First, the liver plays an essential role in lipid metabolism. It is involved in lipid synthesis, storage, and distribution. Lipoproteins, including VLDL, are synthesized by hepatocytes to transport lipids to various tissues. Excess dietary carbohydrates can also be converted into lipids by the liver through de novo lipogenesis and stored as triglycerides. To maintain blood sugar levels during fasting or low carbohydrate intake, the liver can convert stored glycogen into glucose and release it into the bloodstream. In addition, when glucose becomes scarce or limited, the liver oxidizes fatty acids or energy and produces ketone bodies. Next is the muscle cells. Skeletal muscle cells are in charge of using fatty acids for energy, especially during endurance activities. Within the mitochondria, they oxidize fatty acids, producing ATP for muscle contractions. Regular physical activity and training can improve overall endurance by increasing the capacity of muscle cells to oxidize lipids. In conclusion, lipid storage and utilization are important processes in the human body. Adipose tissue acts as a lipid storage center, while hepatocytes and muscle cells are involved in lipid metabolism and energy production. To maintain energy homeostasis in the body, hormonal and nutritional factors tightly control the regulation of these processes.
In this slide, we're going to cover the hormonal regulation of metabolism. We're going to break down insulin, glucagon, and epinephrine, regulates the metabolic processes in the body, specifically in liver, muscle, and adipose tissue. After consuming a high carbohydrate meal, blood glucose level increase, and this causes insulin secretion by the pancreas. The hormones insulin, glucagon are produced by the specialized pancreatic cells called the islets of Langerhans. So alpha cells produce glucagon and beta cells produce insulin. When blood glucose level GLUT2 to transporters allows glucose into pancreatic beta cells were phosphorylated by glucokinase and hexakinase 4 and form glucose 6-phosphate, then enteric glycolysis, the citric acid cycle, and oxidative phosphorylation yielding ATP. Now an ATP concentration increased ATP-gated potassium channels in the plasma membrane, which closes the polarized and membrane. This depolarized voltage-gated calcium channels allowing calcium into the cell, which increase the intracellular calcium concentration. Now as the calcium concentration increases, this is going to trigger the release of insulin by exercises, so insulin will then lower blood glucose level by taking up glucose in the tissue. When glucose level decreases, the pancreatic beta cells will detect this and it's going to inhibit the glucose kinase
reaction and stopping the release of insulin. This feedback is what regulates insulin release. So glucagon is secreted by the pancreatic alpha cells. This occurs several hours after food intake or in between meals because the brain and the tissues are oxidizing glucose for energy. So when the glucagon secreted increases, the insulin release will decrease. So what are the metabolic effects when glucagon is secreted? First, it's going to trigger glycogen breakdown or glycogenolysis in the liver that can yield glucose. Glucagon is going to stimulate glycogen phosphorylase, which is the enzyme that breaks down glycogen to glucose. Glycogen breaks down, it's going to inhibit the glycogen synthesis because we don't need to store glucose. So glycogen synthase, which is the enzyme produced by the glycogen synthesis, is going to inhibit. So referred back to the regulation of the glycogen breakdown and the synthesis, where we covered how this enzyme glycogen phosphorylase and the glycogen synthase is phosphorylated and dephosphorylated, removing and attaching phosphate groups to active and inactivate this enzyme. Now glucagon is also going to inhibit and slow down the glycolysis, and it does this by inhibiting the enzyme PFK1. On the other hand, it's going to activate glucose synthesis by gluconeogenesis. So the formations of glucose molecules from non-carbohydrate source, so pyruvate and lactate, amino acid and cholesterols. Then glucagon is going to increase the FB phase 2 activity. So the FB phase 2 is activity wherein it increases the PEP carboxykinase is decreased pyruvate kinase. So the pyruvate kinase is the enzyme that is responsible for and catalyzing the last step of glycolysis, converting phosphorus phosphate pyruvate, and it does this by triggering the cycling of AMP dependent phosphorylation. Let's discuss the physiological and metabolic effect of epinephrine. So epinephrine is secreted from the adrenal medulla when we're faced stressful situation. This wherein fight or flight response. For example, walking into the room and there is a tiger ready to bite your head off. This is going to stimulate the release of epinephrine and norepinephrine to prepare your body in action and increase the work of epinephrine acts muscle live off. Adipose tissues epinephrine is going to increase your heart rate and blood pressure, so dilating your respiratory passages so you can increase oxygen intake and delivery to muscles. While on muscles, going to increase the glycolysis activity by increasing synthesis of fructose 2,6-phosphate, which is an allosteric activator of PFK1, so you can produce more ATP or energy to produce or to support your muscle. It's going to inhibit, like you're going to convert glycogen to blood glucose. Increasing glucose for energy is also going to stimulate anaerobic degradation of glycogen in muscle by lactic acid fermentation so that we can produce ATP. So collection synthesis activity is going to be activated so we can produce more glucose for energy that doesn't stop. The body requires more energy, so epinephrine is also going to release fatty acids from adipose tissues. So similar to the action of glucagon, it's going to activate hormones and sensitive lipase. Another fact of epinephrine is going to increase glucagon secretion and decrease insulin release. So, to summarize, the epinephrine, it is going to be an activating all the pathway that produces glucose and ATP because we have increased work. We are in a fight-flight situation, and it's going to decrease all pathways and processes such store and so is the hormonal regulation of metabolism. In this lecture, we learned how the hormones, insulin, epinephrine, and glucagon regulates the metabolic process in the body and how it involves the different metabolic states.
Disorders exhibit increased or decreased levels of lipids that cause various health effects in the human body. These types of disorders increase triglyceride, low-density lipoprotein, or both lipid levels. Sometimes called the bad cholesterol, high levels of LDL eventually result in heart disease and stroke. It can be perceived through the following disorders. First is hyperlipidemia. This happens when abnormal high levels of lipids are present in the blood. These are further conceptualized into two branches, based on the type of lipid, which consists of hypercholesterolemia and hypertriglyceridemia, and based on its causing factor comprised of primary and secondary hyperlipidemia. Second is LDL hypercholesterolemia. It is characterized by elevated or high levels of bad cholesterol. This makes fat collect in your arteries, known as atherosclerosis, which puts a higher risk of heart attack and stroke. It can also lead to cardiovascular disease, such as stroke, coronary, and peripheral artery disease. Next is isolated hypertriglyceridemia. This occurs when triglyceride levels are higher than normal and cholesterol levels might be tolerable or not. Next is diabetes mellitus. Continuous health monitoring is a necessity for patients with this disease for their long-term survival. It is a metabolic disease that involves abnormal blood glucose levels. Gaucher's disease. It is a rare genetic inherited disorder. It happens when there is no enzyme that will break down fatty substances. These lipids start to build up in certain organs such as the spleen and liver, which can get very large and will eventually stop working in its normal state. There are 3 types of this disease, including type 1, the most common type, type 2, the form that affects babies, and 3, the kind that consists of skeletal problems, etc. Last is Tay-Sachs disease. Just like Gaucher, it is rare and passed down from generations. If the former builds up on the spleen and liver, this type causes too much lipid in the brain, which destroys nerve cells, causing mental and physical problems. Unfortunately, there is still no treatment for Tay-Sachs in the present. Let's proceed with the impact of diet and lifestyle on lipid metabolism. Some of the aforementioned diseases are the result of unhealthy lifestyles. Mismanagement of food diet, drug therapy, and proper health checkups contribute to the deterioration of one's lipid metabolism. If dietary measures are to be taken seriously, it will help decrease the risk of atherosclerosis and cardiovascular diseases. For instance, a person with high cholesterol levels must adopt a diet low in saturated and trans fatty acids. They should incorporate foods rich in fiber, antioxidants, plant sterols, and stanols. Needless to say, it is much more effective if they comply with regular exercise and maintenance of healthy weight. For prevention, a person must avoid ingesting food with saturated fats, such as dairy products, fat found in animal sources, including chicken skin and red meat, and high trans fat diet which can be obtained from commercially baked cookies and ready-to-eat meals. We will now discuss the medical interventions and treatments for lipid-related conditions. The most commonly prescribed medication for lipid metabolism disorder is statin, which is the initial resort if lifestyle changes are insufficient. It primarily blocks the enzyme that produces cholesterol, hence lowering LDL. Furthermore, it is the most potent class of medicine used for cardiovascular diseases. Other medications include cholesterol absorption inhibitors and bile acid resins. The former functions by lessening the amount of cholesterol ingested, the latter binds to acids in the intestine's bile for the prevention of cholesterol absorption. Fibrates are necessary for the body's production of triglycerides. They may increase levels of HDL, also known as good cholesterol. However, all of this will be ineffective or will resurge once the medication has finished if a patient chooses to be ignorant of their lifestyle and careless of future consequences. In order to enhance the importance of understanding lipid metabolism for overall health, just like other processes in the body, understanding the functions and benefits of lipid metabolism is crucial to one's overall health. As the metabolic center of the body, it can lead to the loss of its normal functioning state, which can lead to worst-case scenarios such as chronic diseases. Lipids are the all-rounder components of your body since they partake in diverse roles. They serve as the structural building blocks for membranous and cellular composition. Additionally, they play as molecular messengers which signal the molecules to perform their tasks. As biomarkers, they act as detectors for early warning systems of the possibility of diseases. Lastly, they provide energy for living organisms, providing more energy than an individual can take from carbohydrates and proteins. As they all say, too much of something is never a good thing. Because lipid metabolism is mainly affected by the food we consume, we must monitor it and adequately take the right amount of it. Excessive amounts of lipids are life-threatening, but so is a lack of dietary lipids. For our body to perform its physical tasks properly, we must first be reminded of our internal mechanisms. Think of it as child's toy, a building block. You can progress towards the higher stages if you haven't completed the base. In that analogy, the lipids, along with other metabolic processes, serves as the base. They are the foundation that builds up the energy for us to function appropriately.
Do ba 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 do.
Sabi ka
Do ba do do do.
Sino ba ang una, di ba?
Abu ba do ba do.
Start na to. Di ba introduction to? Ihati-hati na tayong parts. Ito may introduction din.
Adu ba do ba do.
Kasi sobrang
Adu
kapag
ba do ba do do do. Adu ba do ba do. Adu ba do ba do.
Okay, may lalaway yung tignan. Wala ko sa lakan. Hindi ko siya sa akin pinagputulin. Kasi yun yung
Adu ba do ba do. Adu ba do ba do ba do. Adu ba do ba do ba do.
Kasi dun sa part na yun, napasok kasi yung records na time na ano na vocal pa ako sa ano. Sabihin mo kayo sa ano, like, bibigyan ko lang sa ako. Hindi mo yun,